Okay, we're going to start with what is called interferon. Now, we are still in what is called the second line of defense. So we went through all of this, but we are still under this, the body's second line of defense. And especially things like this where it says antimicrobial chemicals, interferon. This is pretty cool, actually. Interferon is a defense against viruses. Very relevant topic in today's society. There's three kinds. There's alpha, beta, and gamma. But what happens is when a virus infects a cell, and what viruses do, they bind to the outside of a cell, and once they get inside of a cell, they do two things. So showing it right here, look at this. See, here's a virus binding to a cell, and they inject their DNA or messenger RNA inside the cell, and then the cell transcribes the DNA and then translates it. If it's messenger RNA, it just translates it. And there is a stage of a virus cycle where it becomes dormant and part of the genome, the genome of a cell, which they don't do this. But viruses invade cells like this, and they cause the cell to produce other virus particles. You see that? Viral nucleic acid, assembly of viruses, and now they're being released, and that virus is going to try to then invade another cell. So here's what interferon does, and I know this is under complement, but I really didn't have anywhere else I could put it. And make it this big. Interferon is released by a virus-infected cell. So these right here. Chemicals produced by a virus-infected cell that travel to surrounding cells and cause those cells to produce antiviral proteins, preventing the virus from infecting those cells. And that's what that's showing. See, here is supposed to be the release of interferon, and it's causing an activation within the nucleus of a cell. And right there it's showing synthesis of antiviral proteins. So that way it keeps the virus from infecting other cells. Gamma interferon gets released, and basically it causes one of these things, a monocyte or slash macrophage, a phagocyte, to show up and basically destroy the virus-infected cell and the virus inside. So that's actually pretty cool. Complement, and maybe I can, it works pretty well there. So complement. So what is complement? This is another part of the nonspecific second line of defense. A set of 20 regulatory proteins in plasma designated numerically according to the order of their discovery. And yes, if you take immunology, you spend 2 weeks talking about this, so that's not our purpose here. I did post two diagrams. Here's one. Here's adaptive response. We haven't gotten there yet. Here is where we are. Innate immunity and inflammation. And this is an antigen-presenting cell, an APC, a cell that's come in contact with a foreign antigen. The foreign microbe has been destroyed, and its antigens are placed on top of a cell, and it goes around displaying the antigens and thereby causing immune responses. Big long words there, but I just wanted to show you the complement system is all connected to the whole entire immune response during an illness. And it does all these things. Now, I did post another, and I hope this one, it's a super detailed. See the C3b, like it said there, they're sort of numerically numbered in order as how they were discovered. But what do? What does complement do? Complement is mass chaos. When complement becomes activated during an immune response, it increases phagocytosis. And how does it do that? Here's one way. See this opsonization? Basically, these proteins will coat the outside of this microbe. And when it does that, it becomes a target for monocytes and neutrophils. And they engulf it. So it's a pathogen. What else? Well, they do increase inflammation as well, which that's the third one. They cause lysis of foreign pathogens in cells. Same thing. Down here, let's see. It says platelet activation. Well, there it is. See, it's penetrating. It's actually stabbing these cells up here. If you can see that. Down here, it says bacterial killing the iMac, and you see it actually causes, those are little it's almost like a spike, and it's poking holes in the cells. And the third one increases inflammation, and I tried to summarize that because this class is not based on complement, but down here it says interaction with adaptive immunity, and apoptotic cells mean it's killing cells. But basically, it does cause inflammation, and I was hoping maybe it's not really showing it on here. It says pro-inflammatory effect, but it does cause inflammation. The thing about inflammation, it keeps a pathogen from moving around the body. So this ended up being a super detailed diagram, but here are all the DAF, CR1, CFBP, and those are all the names for the little individual proteins, and apoptosis means killing of cells. And there's some pathways. Say it's not our purpose to go through all of these, and down here what it's showing is its complement helps activate other cells as well. So it's just like this, how it's all involved. But they do that, and like I said, this is actually pertaining to interferon. Just didn't fit where I wanted it to. Fever. Fever is body's response to invasion of microbes. Once the immune system kicks in and complement and neutrophils and monocytes start fighting and engulfing and destroying foreign microbes, the phagocytes, they release a chemical called cytokine interleukin 1. That's what causes fever. The old name was endogenous pyrogen, which is a cool name. That means fire maker within. But once that chemical gets released in blood, it causes the hypothalamus to reset the set point. And normal body temp is 98 degrees. That's the set point. That chemical causes, and it depends on how virulent the strain is and what kind of microbe it is, but it causes the hypothalamus to raise body temperature. That's why sometimes people get chills and shiver when they're running a fever, because it's the body's attempt to get muscle contractions to generate heat to match the new set point. And what that does, pathogens that make human beings sick, they prefer to grow at 98 degrees Fahrenheit. That's the temperature they're comfortable with. Fever makes that uncomfortable for them, so it disrupts their life cycle, basically. I said growth cycle there, same thing. I'm going to cover this, and then I'll cover the last part in the last video. So what we just did is go through the first, they're called non-specific defense, first line, second line. This is the third line of defense. There's not going any more, and it's called adaptive immunity. Now this is very specific. This type of immune response is only going to attack the stimulus. In other words, the foreign invader, foreign antigen that caused this whole immune response. These cells, and it's B cells and T cell lymphocytes, they're only going to attack that antigen, that foreign pathogen. I still have the picture up. This guy says pathogen, and I keep saying foreign antigen. Antigens are proteins on cell surfaces, and anything that's a foreign antigen is a pathogenic or foreign body. Okay, and so let's go from here, and then I'm going to stop after the little description about how primary and secondary works. But there's two parts of this. There's adaptive immunity, and it has two parts. It has the cell-mediated immune response and the antibody or humoral-related immune response. This one's T cells, this one's B cells. So just get familiar with the terminology. A foreign antigen is a foreign microbe. Okay, it's a pathogenic thing or something the body doesn't recognize. Like it says there, our microbes are substances that can cause an immune response. So here's what happens. If something has made it through the first and second lines of defense, at some point, and I'm just using this here, see this down the bottom? It says cell recruitment, and the other picture. At some point, a foreign microbe is going to come in contact with a T cell and a B cell in the bloodstream. And that's where this comes in. And that is what, now you're going to get what is called an adaptive immune response. So here's some terminology. Primary immune response is the first exposure to a foreign antigen for these B cells and T cells. The first exposure has a longer lag time. It has less antibody, in other words, less B cell and T cell production, and B cells produce antibodies. And we'll get to those in the last video. And so the very first exposure, the lag time takes forever. The lag time is the time it takes for the body to develop an immune response. And the very first response, it takes longer, and you get much less production of the cells that are going to fight the infection. The secondary immune response is the second exposure to the exact same antigen. So if a person gets sick, if somebody had smallpox back in the day and it did not kill them, which it had a very high mortality rate, if they got exposed to smallpox again, if they survived the first time, then they're going to have what's called a secondary immune response. And the second exposure is very fast, and the reason for that is there are things called memory cells that we'll get to in the last video that are produced after the very first exposure. And those memory cells are stored throughout the lymphatic system, and they're memory cells very specific for that antigen. But the second response, much shorter lag time. I'm going to add that actually. It is much faster, but it's a shorter lag time, and more production of antibodies and T cells. This is a great graph. This will be the last thing in this video. This is supposed to represent the primary immune response. On this side over here, you can just, it says plasma antibody concentration. You can also think of that as also T cells. And you see, look, this is the days down here, days after exposure. Look, this thing peaks out, what, about day 22? If a person got smallpox, it was fatal within 4 days. So that's why I had a high mortality rate. And it takes, long time to get going. It peaks at 22 days, and you can see there, this axis is showing by numbers how high the numbers got as far as the army to attack this foreign antigen. This is the secondary response. Look, almost immediately, 2 days? And then it peaks right about day 10. So that's twice as fast. But already by day 5, it's already as high as it took 22 days to get here. And you can see the numbers are much greater. And this is the entire basis for getting vaccinated. If a person gets vaccinated, they get injected with a dead or weakened pathogen, and it might even cause some symptoms. Unfortunately, there have been people historically that have died from getting immunizations from it, but it doesn't happen very often. But what ends up happening is you get injected with this weak or dead pathogen. You get a primary immune response. Your body's going to develop memory cells and some immunity to that very specific pathogen, and then if you actually get exposed to the real thing, you're going to get a secondary response, and then chances are you're not going to get it again. Okay, and that has to do with these types of cells, and I'll start there next.